Next, we want to move on to spectroscopy, right? And the first one we're going to look at is actually one that's extremely tied closely to absorption, which we just talked about. It's called IR spectroscopy, and the IR stands for infrared, right? Which, remember, is a certain wavelength of light. Infrared, it's slightly lower energy and higher wavelength than visible light. Okay, so this is one very common type of spectroscopy that many of you have likely seen before. The main principle is based on the absorption we just covered, specifically for the IR region. You can also do UV visible light spectrums, but those I think are a little bit less common, and those are not used necessarily for identification in this way. They're more used to identify whole molecules that have known UV visible light spectrums, okay? But this is something that they would ask you more on like an MCAT, this type of spectroscopy, and this is one that you probably learned in organic chemistry class. So you can see here, you have the infrared spectrum for hexanol. It has this transmittance, it has this wave number, which is not wavelength, but wave number. Transmittance is basically how much it passes through, essentially. And you have this little region, which is not really super important. This is like the base zone, I guess, and you don't really pay attention to this region almost ever. It's really starting from like, I think, here and left that you are really looking at. This region is kind of like the background noise, I guess, kind of. It'll be there like for most of the spectrums that you see. And what you're looking for is these peaks, right? This one, and this one. And really what you're looking at is where on the x-axis they are. So where does this correspond to? This appears to be about 3,300. This is about 3,500. This is about 2,800, right? Okay. And you're also looking at the size. This is a little bit of a thicker, wider peak, whereas this is a very short peak. This is a somewhat short peak, but very intense, as in it goes down a lot on the y-axis. So just keep these descriptions in mind. And this topic is very much, you have to just memorize it, and that's about it. Just memorize and apply. There's not much thinking about it. And I will show you here, this is what you have to memorize. This table is very important. This will basically describe what the peaks for every single type of bond that you will see look like. So a benzene ring is 600, will have a wave number about 1600 and 1500. C double bond N will have 2200. C double bond C, it's basically the same thing, 2200. C triple bond C, sorry. C double bond C will be less, about 1650. Same thing with C double bond N. C double bond O will be about 1750s, but it'll have a very strong peak, like you can see here. It's a very intense peak, right? Goes all the way down essentially, whereas these are more mediums. These will go like halfway down. So pay attention to this. This one, OH alcohol, this one's pretty important. Broad. These guys, the OHs, they're very broad, as in they're very wide. Okay, you can see that's what it is here. Okay. And again, this is the cutoff for what it's actually important. This is kind of like the background noise you're not really paying too much attention to. As you can see here, it's like a maybe, but really you'll be able to identify what a molecule is based off of these two peaks alone. Okay, and in this case, it's ethanoic acid. Okay, so again, this is just straight memorization and application. There's not really much else to do here. Unless, yeah, you just have to memorize, and that's all you can really do in this situation. If you don't memorize it, your chances of getting a question, if this appears on MCAT, your chances of getting it are pretty low, I think. But luckily, it doesn't appear, I think, too often, but I think it does consistently appear on the MCAT, but not very much. So you'll probably see like a question or two on every MCAT on something like this, like the IR spectroscopy, but probably no more than that. Next up is proton NMR, and this one is the exact opposite, where it's pretty much very little memorization. but it has a lot of actual like thinking and reasoning and like solving it out and writing it on paper, I think is very helpful for this type of problem. Okay. So proton NMR, as the name suggests, is you're measuring the hydrogens, the H pluses or protons in a molecule, and you use that to kind of identify what the molecule is or what the structure is, roughly. So you can see here, this is color coded. These three blues apply to this. These three greens apply to this, and these two reds apply to this. Okay. Now, what are all these peaks and what does all this mean? So there's two things that you have to look at for proton NMR, and only two things. Firstly, the number of neighbors, and secondly, the number of equivalent hydrogens or equivalent protons. And we'll cover that here, and I'll come back to this screen in a second for you guys to see, but I want to talk about this. So the n+1 rule, this is the neighbor rule. This rule states that the number of peaks in the NMR will correspond to the number of neighbors proton has, which is n, and one extra, so n+1. Remember, a neighboring proton refers only to protons on neighboring carbons, not protons on the same carbon. Okay, so let's see. Neighbors. We'll start with this blue one first, this hydrogen. How many neighbors does, and this corresponds to this peak, is color coded blue. So how many neighbors does this blue hydrogen have? Well, neighboring carbons. So it's on this carbon. So we're looking at this carbon. Now, how many neighboring carbons does this carbon have? Well, to the right, there's nothing because it's an N carbon, but it has to the left here. Okay, has this one right here. So this carbon has two protons on it. Okay, that means this blue hydrogen has two neighbors, has two neighboring protons, right? Because it has this one and this one. These, remember, don't count. These are on the same carbon. Has to be on neighboring carbons. Has two neighbors, therefore the n is 2, n+1 is 3. Therefore, this blue line should have 3 peaks. So 1, 2, and 3. 3 peaks, exactly. Okay, simple. Now let's talk about this one. Or sorry. Talk about this hydrogen, and I'll go through this one a lot quicker. How many neighbors does it have? This carbon has only one carbon neighbor. This one, because this is an oxygen. Oxygens have nothing, unless it's in alcohol, in which case it does count. Okay. Remember, alcoholic hydrogens also count. Carbons. This neighboring carbon has 3 on it. So that means this red hydrogen has 3 neighbors. n+1, 3 + 1, 4. So that means the red one should have 4 peaks. 1, 2, 3, and 4. 4 peaks, right? This one, oh, sorry, again, sorry. This green hydrogen, how many neighbors does this carbon have? Well, it has one neighbor. It's an N carbon, it only has one neighbor, but this carbon has no hydrogens on it, no protons, it only has 2 oxygens. Therefore, this green hydrogen has 0 neighbors. So 0 + 1 is 1, so it's just 1 peak. Okay, hopefully that clears up the neighbor rule. The n+1 rule. That's the first half. The second half is the size/intensity of the peaks. The size/intensity of the peaks is determined by the number of equivalent protons that peak represents. As a rule, all protons on the same carbon will always be equivalent to each other. But protons on different carbons may also be equivalent if the molecule is symmetric. And I'll give you an example of this. For example, if I had a benzene ring with an OH, well, let's say actually for simplicity, with a double bond O, okay? We had this molecule, and you can see that each of these has hydrogens on it. I'm not going to draw each one, but all of them have 2 hydrogens on them. Obviously, this hydrogen, and I'll actually do different colors. This hydrogen and this hydrogen are equivalent because they're on the same carbon, right? So they're exactly the same, right? It doesn't matter. They're basically the same hydrogen because they're in the same position. They get the same amount of spin and shielding and all of that, which is principle behind NMR, but you don't really have to know that. The principle is like it provides a spin and it gets shielded by its neighbors, but again, not important for the MCAT. You just have to read the graphs and draw the molecules based on the graph. Okay, so these two are the same, right? Because they're basically in the same position. But this molecule down this line is symmetric. Therefore, these two hydrogens are also equivalent to these. So all four of these hydrogens are essentially the same hydrogen. They're in the exact same positions. They're equivalent, right? So there's actually 4 equivalent of red hydrogens. Now let's do blue. Obviously, again, these two are the same. These two hydrogens are the same because they're on the same carbon. But again, they're on symmetry. So these are also blue. And lastly, green, just these two. And because they're on the point of symmetry, they don't have any anything to be symmetrical with. So it's just that. Okay. So the reds are equivalent, the blues are equivalent, the greens are equivalent. That works because there's symmetry. If you had, for example, something like this, where it was just like a double bond O, and then you had a F here or something like that, right? Like a fluorine or something. And then you had carbon here. There is no longer a line of symmetry. So these are now different. So these two are still reds, but now these two are greens. And these are also not the same. So these are now blue and purple. Okay? Because again, no more symmetry. The symmetry has been broken. So the hydrogens here are now all different. Okay? So only when there's symmetry, carbons that are symmetrical can be equivalent. Now let's talk about what it looks like on the graph. So again, I mentioned the size and intensity. So it's color coded here, just like I did. So all of the blue hydrogens, there's 3 blue hydrogens that are all equivalent to each other. So that means these 3 peaks have to have high intensity, let's say, or the intensity equal to 3 hydrogens. These only have 2 hydrogens, so it has lower intensity. These 3 greens are, again, 3, so it will have higher intensity. And the intensity between these two should be equal, right? But you notice that this peak is big, this peak is smaller, but this has 1 peak, this has 3 peaks. Really, if you put these 3 peaks together on top of each other, it would equal this one big green peak, but yeah. Okay. Remember, these should be equivalent in terms of intensity. Okay. All right, moving on. And that's all it is, proton NMR. So there's these two things you have to keep in mind: intensity with equivalent carbons, and number of peaks, which is the N+1. Okay. Remember, neighbors, neighboring carbons, and equivalent carbons. That's really all. Okay. Next, let's move on to carbon NMR. Carbon NMR is similar to proton NMR in that it helps identify the different carbons that exist in a certain compound, but there are some distinct differences. Main difference is that in carbon NMR, rather than the number of peaks, we look at the position of the peaks on the x-axis to determine what kind of carbon bond exists. Carbon NMR, if I had to describe it, it would be a combination of proton NMR and IR spectroscopy. That's the best way, in a sense, to describe it. And this is what it looks like. So depending on what kind of carbon bond you have, whether it's carbon double bond O, like a ketone, an ester, benzene ring, a double bond, a triple bond, a bond with a halogen, or just a regular single bond, has a different position on the x-axis in parts per million. Again, this is another chart that you have back to just memorize it, and that's it. It's another chart that should be memorized, and be careful. There are not many patterns between this list and the previous one for the IR spectroscopy. Remember, this list also had some bonds, but there's not too many patterns between these. Maybe if you see some, go for it, but just be careful not to get mixed up between those two lists. These two lists are separate. They should be memorized separately. Okay. Another important difference between proton NMR and carbon NMR is that you are allowed to ignore the intensity in carbon NMR for the MCAT. It is not related to the number of equivalent carbons like it is in HNMR. So even if you have multiple equivalent carbons, they're not going to increase the intensity. That's not how it works in carbon NMR. It's based on something else. It's based on like protons, but it's way too complicated for the MCAT, and they don't ask that usually. Okay, so this is what it would look like. This is what a carbon NMR would actually look like, and it's labeled here. So C1 is here, C2 is here, and you can see that it's the ester, right? Then C3 and C4 are relatively close to each other. This one is this have an O bond, so it's a little bit more to the left, whereas this is just a carbon-carbon bond for 3, for 4, so it's just, yeah. Okay. Basically, again, just memorization and application. Carbon NMR, I think, is the least common one out of the HNMR and IR spectroscopy. It's the least common one, but sometimes it does appear. Another thing to note, you usually use C13, which is the radioactive form of carbon for carbon NMR, so just keep that in mind. If you see C13 NMR, that means just carbon NMR. Next up, let's talk about mass spectrometry. Mass spectrometry is a technique that separates molecules based on a mass to charge ratio, after which they can be ordered in relative abundance, as seen here. This can be used for isotopes, which is shown here, right? Remember, isotopes, elements exist in different masses. Since the atomic form has the same charge, you can essentially eliminate the charge ratio. So you can just order them based on mass because they all have the same charge. So the mass to charge ratio will just be essentially based only on mass. There's only one variable. Okay, and then you can just order them based on mass. And then the relative abundance you can just measure out, right? Once you have them separated. Or it can be used for mixtures of very similar molecules with uniform charge. Again, remember, unless you have a uniform charge, it's a little bit hard because there's two variables, so it's a little bit harder to separate into order, as in terms of a separation. But if they have a uniform charge and they're very similar, you can essentially separate them from a mixture. Okay, and that's more for biology than chemistry. The mixture and the isotopes is more obviously for chemistry.